All right. So like Adam was saying, getting biomass, you know, how much fish do we have in this lake or how many mussels in this mussel bed has been a goal of the eDNA field, you know, really since it started. And so this talk is just kind of where we are right now with trying to do that. And I think it's helpful to start by making the distinction between relative biomass or absolute biomass. So if you're measuring relative biomass, that means you can say there are 6 times as many silver carp at site A relative to site B. And eDNA measurements can already do this. If you have a good quantitative assay and you have similar environmental conditions at different sites, then the eDNA concentrations do correlate to biomass. There have been demonstrations of this in the field with many different species, so I think we are there. But of course, for each species, there's some calibration that goes into that. And then there's the question of absolute biomass. So that's where you're going to say there are 10,000 kilograms of silver carp living in this tributary. It would be great to be able to say that, but really absolute biomass is very difficult to measure, even for traditional sampling techniques. And traditional methods like side scan sonar and mark recapture and catch per unit effort surveys will often put their results in terms of biomass, but I would argue that traditional measurements are also truly measuring relative biomass because there are biases associated with every different capture technique. And so I think it's really useful to remember that distinction. One situation where we truly can have absolute biomass is when we've got the fish in the lab and we can weigh it, and count the individuals. So, and this is where our lab has done a lot of experiments to look at the performance of eDNA in a situation where we control the environmental variables and we really do know the absolute biomass that we are working with. So here's a very detailed scientific diagram of our basic experimental setup. We start with well water flowing into a fish tank, and we know the flow rate, so we measure that in liters per hour. We know how much fish is in the tank, how many individuals, and their total weight. In the outflow, we can take our measurements, and that's where we find the fish DNA. And we measure that in copies per liter. And then if we multiply the eDNA concentration by the flow rate, we get an eDNA shedding rate or flux in copies per hour. So that's the actual number of copies of the target DNA that the fish is putting in the water over time. And that number, the flux of eDNA, is what is related to biomass. So there are a couple ways of measuring what that amount of eDNA is. A lot of us use targeted approaches of quantitative PCR or digital PCR. Our lab uses the Biorad digital droplet PCR system, and that seems to work really well. So that will give you the presence and relative abundance of one target species. And the results are quantitative, but of course the direct measurement that it gives you is the concentration of the eDNA. So it is an indirect measurement of the relative abundance of the target species. And then next generation sequencing or metabarcoding can also give estimates of abundance. And I know some labs have had pretty good success in using this in a quantitative way. Definitely, it's been shown that metabarcoding can give you relative rank abundance of species. But the quality of the quantitative information depends on calibrating the primer set and how much primer bias there is for the species that you are interested in. And all primer sets will have some bias. So the quality of the results will depend on how well those primers work for your particular set of target species. So here's an example from our lab using freshwater mussels. This species is called the mucket. And so here on the x-axis, we have the total biomass of muckets that we put in our tanks, and that's on a log scale. And then on the y-axis, we have the eDNA shedding rate. And again, this is on a log 10 scale, and it's in copies per hour. And it makes a nice straight line. You can see there's some variation around the line, and that seems to be an inherent feature of eDNA measurements, that eDNA is clumpy in the water, and there's a lot of variation from sample to sample, even in a controlled system like this. Also, I should mention that these mussels are all in the same life stage, and they're fairly similar in size. So the variation in biomass is from the number of individuals, from how many mussels we put in each tank. So we would not expect any effects from different life stages in this experiment. And we're working here with a relatively small range of biomass. You know, there's a less than 10-fold difference here between our low treatment and our high biomass treatment. So then here's another experiment of a similar type where we were able to get a little wider range. So this is silver carp and bighead carp. The bighead carp are the circles and the silver carp are the triangles. We had two size classes. We called them juvenile for the smaller size class and sub-adult for the larger size class. And those are the smaller symbols are the juvenile and the larger symbols are the sub-adult. Again, we have the absolute biomass on the x-axis, and again, we got that by actually weighing the fish, so this really is absolute biomass. And on the y-axis, we have the eDNA shedding rate in copies per hour, and both axes are on a log scale. So here we have a much wider range of biomass, you know, over 2 orders of magnitude. And again, we see a pretty straight line here, which is very encouraging for this whole field. And then when you have data like this, here's where you can look at the numbers and say, okay, look at that, you know, 10 to the 3 grams of fish over at the right-hand side of the x-axis. You can look up to where the points are and read across. Okay, so that means for every kilogram of silver carp or bighead carp, I get approximately 10 million copies of their mitochondrial DNA per hour in the water. So that's where you could use your eDNA flux measurements to give you an estimate of absolute biomass. And it can be instructive to do that in order to make comparisons with the estimates that you get from traditional gear to find out if you're even in the same ballpark. But I would just urge that when you do that, remember that both techniques are really doing relative biomass. And so if your environmental conditions change, the measurements that you get are likely to change. And that's because Environmental and physiological factors can change the relationship between eDNA flux and biomass. So some of those factors, which you might not always know what they are in the field, are food availability. That is, the more that a fish eats, the more it excretes, and the more DNA is likely to be in the water, even for the same amount of fish. When the temperature goes up, kind of just overall activity and metabolism increases, and the amount of eDNA shed can increase. There can be differences in eDNA shedding rate with life stage, and this is some of Matthew Yates' work, that smaller fish tend to release more eDNA per gram than larger fish. Reproductive activity will put more DNA in the water, from gametes and just from the reproductive activity in general can put a lot of DNA in the water. Changes in pH and differences in microbial activity can change the degradation rate of the DNA, and also stress on the fish. If you go out and chase the fish around and catch them in nets, there will be more DNA in the water, even if you didn't actually catch any fish. All right, so here's some actual data on some of those factors. So here's food availability. So in this experiment, we looked at big head and silver carp. Again, these are the same juveniles and subadults that we studied for the biomass experiment. We fed them a diet with a low amount of algae, a high amount of algae, brine shrimp, and then no food at all. And again, on the y-axis, we have the shedding rate in copies per hour and on a log scale. And consistently, the algae diets had about an order of magnitude more eDNA, more copies per hour than the no food. And there's also, again, a lot of variation in this data. When we looked at temperature in the lab, we actually did not see any significant differences in shedding rate. But this was over a kind of small temperature range from 19 to 31 Celsius. So the fish were getting the same amount of food at all these temperatures. But we did see an effective temperature when we did a field experiment, which I will show here. So this was an evaluation of the kind of eDNA consequences of taking a lot of silver carp out of Creve Coeur Lake, which is a floodplain lake that is here in Missouri near St. Louis. It had a lot of silver carp in it, and so Dwayne Chapman's team and Fish and Wildlife and our state Missouri Department of Conservation worked together to chase all the fish into one corner of the lake and then systematically seine them out. So the goal was to remove as many of these silver and bighead carp as possible from the lake. And we periodically, before the removal and after the removal, took eDNA samples from a grid over the whole surface of the lake to see what would happen to the eDNA concentrations. So here I have our results plotted. Again, copies per liter is on the y-axis, but here note the x-axis is temperature. So the blue dots are before the carp removal, and the orange dots are after the removal. And I've just run a regression through those 3 dots that we have before the removal to show that it does look from those 3 points like there is a relationship between the concentration of eDNA and the temperature. And again, all three of those points are before the removal, so there were the same number of fish in the lake for all three of those. So here at the highest temperature of the three was our first sampling time at the end of October. And then here's the end of November. It was a little colder and we saw less DNA. Again, in January, it was even colder. That was about as cold as the lake got. And we had, again, even less eDNA. Immediately, well, 3 weeks after the big removal, that was the most DNA that we had measured, even though the temperature was still pretty cold. And that's because of the effect of the fishing effort and the stress on the fish and carcasses and injured fish and possibly diseased fish being left in the water. And then about a month after that, we measured again, and that's where we could actually see the effect of taking all those fish out of the lake. That was the lowest amount of DNA that we measured. So in order to answer the question that we're actually interested in, so what proportion of fish were actually removed in that big fishing effort, we need to compare the before and after points where we have the most similar environmental conditions. And so that would be the November sampling versus the April sampling. And just looking at those points, it looks like there's about 80% less DNA in the water, after that fish removal. And that number is in pretty good agreement with what the traditional sampling found. So then again, Matthew Yates has introduced a nice improvement to this whole concept by introducing allometric scaling. So you can improve biomass estimates by not assuming that every fish in every size class will have the same DNA shedding rate. So here in these three graphs, we have density or biomass or the allometrically scaled mass from traditional sampling. And on the y-axis, the eDNA concentration that was measured in copies per liter. So just looking at density, that's just the number of individuals. And that gives you a pretty good positive relationship. Looking at biomass, the total weight of all the fish caught gives you a little better relationship. And then when you scale it, that relationship tightens up quite a bit. So that's going to be, I think, a really useful technique to take into the field where we know that our natural populations are made up of individuals of all size classes that probably have all a little bit different eDNA shedding rates. All right, so now I wanted to look at the effects of spawning on eDNA concentration. So again, we're looking at data from silver carp. So here on the x-axis is date, and the green line goes with the right-hand y-axis, and that's the hydrograph of the Missouri River at Boonville, Missouri, which is just west of Columbia. So that's the discharge in cubic feet per second. Then the blue points go with the left-hand y-axis. That's the eDNA flux in copies per hour. So you can see the eDNA flux just kind of hovers around. It's pretty low until we get to the end of May. And then when the hydrograph goes up, the fish spawn, and there's suddenly a lot more DNA in the water. And notice it's not constant. So the DNA associated with spawning kind of comes down the river in pulses. So we were able to measure 3 peaks and 2 valleys during that time of spawning, during the high water. So if you're trying to measure the abundance of these fish, it's that time before spawning that you are interested in. But if you just want to detect the fish, then during spawning is a great opportunity to get a higher concentration of DNA and maybe get a detection even if they are rare. So some challenges for this field are the transport of the DNA. So if we're looking at freshwater mussels, we might ask, well, how close to the mussel bed should we take our water samples? If you're studying fish, that's a more difficult question because the fish are swimming around. And then related to transport is the degradation rate. You know, how long does eDNA last? And in a flowing system, how far downstream will it move during the time before it degrades? Another challenge for this is field calibration. So if we're trying to see how well our eDNA measurements are working, we would really like to compare them to absolute biomass in the field, but the only methods that we have for measuring biomass in the field are, you know, the traditional measurements, which truly are also only giving relative biomass and have their own variation associated with them. Another challenge is that eDNA is generally at low concentration in the water, and so this is going to be easiest to do with the more common species, and obviously harder to do with more rare species. And then, of course, contamination is, you know, always the worst problem for eDNA measurements. So some data that relates to these challenges. Here's a lab experiment that we did on eDNA degradation. And again, this was in controlled conditions in the lab with a high starting quantity of DNA. So here we have copies per liter, again, on the y-axis on a log scale, and time in hours on the x-axis. And the concentration of eDNA is following an exponential decay curve. And the half time in this experiment was 7.5 hours. So that means the concentration of DNA in this experimental system is dropping by twofold every 7.5 hours and approximately tenfold every day. And then the challenge of measuring DNA at low concentrations, there's a recent paper by Mary L'Esperance and Karen Helbing's lab, which used some data that was produced by Katie Klimas, who's in my lab, and a lot of other researchers that collaborated with us from the Gedwig group. So all of the labs that contributed to that study measured their limits of detection in an experiment with synthetic DNA with 24 replicates per sample of these standards at known concentration. So in the data that I'm showing here, this is from our lab. I think it's one of the silver carp assays. And the copies per reaction that's on the x-axis was measured with digital droplet PCR, and then the percent detection on the y-axis was measured with qPCR. And the orange dots are our measured data, and the blue dots are the theoretical prediction from the Poisson distribution, which is it's a binomial distribution that predicts the frequency of events given a number of occurrences. And I think it's really beautiful how well those two lines match up. So Mary L'Esperance has done a statistical treatment to allow us to use the percent detection that we get at these low concentrations where we're not actually getting amplification in every replicate. The percent detection can be converted into a concentration number. So this is a great way to push our limits of detection and our limits of quantification lower and be able to get more useful data at these low concentrations where our eDNA measurements usually are. And then finally, I wanted to look at just one example of metabarcoding and how it does with relative or rank abundance. So this, now we're back to freshwater mussel data. So here in each of these tables, it's a different mussel bed. The species are sorted from most abundant to least abundant based on the visual survey, which is the left-hand column in each group. And then the right two columns are two different metabarcoding markers, CO1 and ND1. So you can see the visual surveys actually did a little better than eDNA in getting longer species lists in general, but we did have a couple species at each mussel bed that were detected only with eDNA. And the eDNA measurements, the read counts tended to be kind of dominated by one or two very abundant species, or I guess one or two very abundant reads, and those could be the most abundant species, or they could be the ones that amplified most efficiently with these primer sets. But overall, the red, yellow, and green colors do tend to kind of agree with the visual survey. And this was, this experiment was not meant to get quantitative information. We were really just looking to detect mussels. So this treatment of the data was kind of just to see how it did with getting at relative rank abundance, and it did okay. So with some calibration, I think you can get some good information on abundance from metabarcoding. So in conclusion, yes, quantitative eDNA monitoring can estimate relative biomass among similar sites. And it really helps to have some lab experiments to go with your field data so that you understand the eDNA shedding rate of your species and the different physiological factors that might change that shedding rate. And of course, those different factors include temperature, food availability, reproductive activity, and probably more. And I think that this field continues to move in a really good direction, and the precision of these estimates will only improve as the field continues to mature. And with that, I'd like to thank my lab and everyone who helped us and our funding agencies, and I will take any questions.